Welcome to DreamLab Knowledge, where we explore the profound mysteries of science, nature, and the hidden worlds that shape our existence through calm, contemplative storytelling. Tonight, we journey inward to discover a universe that exists not in the distant cosmos, but within your own body. A universe teeming with life, composed of trillions of organisms that outnumber your own cells, working ceaselessly in ways both subtle and profound to keep you alive. This is the story of your microbiome, the invisible ecosystem that makes you who you are. The hidden universe inside you, the human microbiome. Close your eyes for a moment and imagine that you could shrink yourself down to microscopic size, small enough to walk upon the landscape of your own skin. Picture yourself standing on this alien terrain, looking out across a world as varied and complex as any continent on Earth. Before you stretches an environment of remarkable diversity. Valleys carved between individual skin cells plunge down like canyons. Hair follicles rise like massive towers, their roots descending deep into the dermis below. Sweat glands appear as great caves from which moisture periodically floods the surrounding terrain. Oil glands secrete their lipid-rich products, creating slick plains and pools. This is not science fiction. This is your body as it truly exists at the microscopic scale, a world unto itself, home to approximately 39 trillion microbial cells. To put this number in perspective, if you could line up all the microbes in your body end to end, they would stretch for millions of miles. If you could somehow separate them all out and weigh them, they would collectively mass somewhere between 1 and 3% of your total body weight. For an average adult, that translates to roughly 1 to 2 kilograms, 2 to 5 pounds of living microorganisms residing on and within you at this very moment. These organisms include bacteria, archaea, fungi, viruses, and countless other microorganisms that have evolved alongside humanity for millions of years, perhaps even longer. They live on your skin, coating every square centimeter of your body's 2 square meters of surface area. They inhabit your mouth, forming complex communities on your tongue, your teeth, your gums, the insides of your cheeks. They colonize your nasal passages, your respiratory tract, even your lungs, once thought to be sterile environments. They establish thriving populations in your digestive system, from your stomach through the winding 23 feet of your small intestine and into the 5 feet of your large intestine, where they reach concentrations that stagger the imagination. Together, these organisms and their collective genetic material comprise what scientists call the human microbiome. The term was coined by Nobel laureate Joshua Lederberg in 2001, though the concept had been gestating in scientific thought for decades before that. Lederberg chose the word deliberately, combining micro for small and biome for ecological community. The human microbiome represents nothing less than a collection of distinct ecosystems, each adapted to the unique environmental conditions of its particular body site, each playing vital roles in maintaining your health and sustaining your life. For most of human history, we knew nothing of these microscopic companions. We couldn't see them, couldn't measure them, couldn't begin to understand their influence on our lives. The very concept of life forms too small to see with the naked eye didn't exist. Then, in the late 1600s, a Dutch merchant named Antonia van Leeuwenhoek, who ground lenses as a hobby, created microscopes of unprecedented magnification. When he turned these instruments towards samples from his own mouth, from dental plaque and saliva, he witnessed something no human had ever seen before: tiny creatures, which he called animalcules, swimming and moving in ways that left no doubt they were alive. Van Leeuwenhoek was astonished. He wrote to the Royal Society in London, describing his observations with a mixture of scientific precision and childlike wonder. He noted different shapes of these animalcules, different patterns of movement. He observed that cleaning his teeth reduced their numbers. He even checked samples from different people, finding that the quantity and types of animalcules varied from person to person. In those primitive observations lay the seeds of discoveries that would take 3.5 centuries to fully develop. But van Leeuwenhoek couldn't have known what he had stumbled upon. He couldn't have imagined that these tiny creatures weren't mere curiosities, but essential partners in human biology. He couldn't have foreseen that understanding these organisms would require revolutions in genetics, molecular biology, computational science, and ecosystem ecology. He opened a door, but humanity would take generations to fully walk through it. The numbers that redefined us. In the early 2000s, as microbiome science began to emerge as a distinct field, scientists needed ways to communicate the significance of microbial life to the public and to other researchers. One figure became particularly prominent, repeated in countless papers, textbooks, and popular science articles. The human body contains 10 times more microbial cells than human cells. This 10-to-1 ratio became a kind of shorthand for the importance of the microbiome, a startling statistic that forced people to reconsider their relationship with the microbial world. The origin of this number traces back to a 1972 paper by microbiologist Thomas Luckey, who estimated bacterial numbers in the gut using what he called back-of-the-envelope calculations. These calculations were rough approximations based on bacterial densities in stool samples and estimates of gut volume. For decades, nobody seriously questioned these numbers. They became accepted fact, cited again and again, building a scientific edifice on what was essentially an educated guess. Then, in 2016, a team of researchers decided to revisit these estimates using more sophisticated methods and updated knowledge of human biology. They carefully calculated the number of human cells in an average adult male body, accounting for every cell type. Red blood cells, which are far more numerous than any other cell type. White blood cells, epithelial cells lining organs and surfaces, muscle cells, fat cells, neurons, and all the rest. Their total came to approximately 30 trillion human cells. For the microbes, they focused primarily on the gut, where the vast majority reside. Using contemporary measurements of bacterial densities in different sections of the gastrointestinal tract, combined with accurate anatomical measurements of gut volume, they calculated approximately 38 trillion bacterial cells in the colon alone. Add in the bacteria from all other body sites, the skin and mouth and respiratory system, and the total reaches roughly 39 trillion microbial cells. The ratio wasn't 10-to-1, it was closer to 1.3-to-1. Some interpreted this as a demotion of the microbiome's importance. After all, we're not as outnumbered as we thought. But this perspective fundamentally misses the point. Even at 1.3-to-1, you are sharing your body with tens of trillions of organisms. The revision doesn't diminish the microbiome's significance. It simply makes the numbers more accurate. More importantly, the numbers tell only part of the story. Because while microbial cells may not outnumber human cells by an order of magnitude, microbial genes vastly outnumber human genes. Your human genome, contained in the nucleus of almost every one of your 30 trillion cells, comprises roughly 20,000 to 25,000 genes. This is the complete instruction manual for building and maintaining a human being, encoded in approximately 3 billion base pairs of DNA. Your microbiome's collective genomes contain somewhere between 2 million and 20 million genes, depending on which microbiome you're examining and how you count. That's 100 to 1,000 times more genetic information than your own genome provides. Think about what this means in practical terms. Every gene codes for proteins, for enzymes, for molecules that perform specific functions. Your microbiome gives you access to a vast library of genetic capabilities that your human genome alone simply cannot provide. Consider carbohydrate digestion as one example. Plants, which humans have eaten throughout our evolutionary history, contain thousands of different types of carbohydrates with complex chemical structures. These include cellulose, the structural component of plant cell walls. They include hemicelluloses, pectins, resistant starches, inulins, fructans, and countless other polysaccharides. Your human genome codes for fewer than 20 enzymes capable of breaking down these various carbohydrates. Without help, most plant material would pass through your system undigested, its nutrients locked away and inaccessible. Your gut microbiome solves this problem. A single bacterial species, Bacteroides thioomicron, possesses more than 260 carbohydrate-digesting enzymes, each specialized for breaking specific chemical bonds in specific types of polysaccharides. Hundreds of other gut microbes possess genes for additional enzymes, creating a collective biochemical toolkit of astonishing sophistication. Together, they can break down virtually any plant polysaccharide you might consume, releasing nutrients that would otherwise remain unavailable. It's as if you were born with just a few Allen wrenches for working with the world, but your microbial companions brought entire hardware stores full of specialized tools. And then there are the viruses. While bacterial cells get most of the attention in microbiome research, viruses may actually be the most numerous biological entities in your body. Most of these are bacteriophages, viruses that infect bacteria. Estimates suggest viral particles in the human body may outnumber bacterial cells by 5 to 1 or even more. That's potentially 200 trillion viral particles, mainly bacteriophages, residing in your gut and other body sites. These viruses aren't just passive passengers. They actively shape microbial communities, infecting bacteria, killing some, transferring genes between others through a process called transduction. They influence which bacterial species dominate at any given time, acting as population control agents that prevent any single bacterial strain from becoming too abundant. They contribute genetic material to the collective metagenome, adding yet another layer of complexity to this hidden ecosystem. The sheer scale of microbial life within and upon us forces a fundamental philosophical question. What does it mean to be an individual? Where do you end and other begin? The traditional answer that your skin marks the boundary of self suddenly seems inadequate. Your body is not a fortress keeping the world at bay, but rather a complex habitat supporting diverse communities of life. You are not a singular organism, but what some scientists have called a superorganism or holobiont, a walking ecosystem in which human and microbial cells work together in ways so integrated that separating them becomes meaningless. This challenges deep-rooted assumptions about biological individuality and identity. If your health depends on maintaining healthy microbial communities, if your metabolism is partly determined by which microbes you harbor, if your immune system requires microbial education to function properly, if your brain's chemistry is influenced by bacterial metabolites, then in what sense are you a single unified being? The question has no easy answer, but it fundamentally changes how we must think about human biology, health, and disease. The geography of the body. Just as Earth's surface can be divided into distinct biomes, tropical rainforests and Arctic tundra, coral reefs and grasslands, deserts and wetlands, your body presents distinct microbial habitats, each with its own characteristic community of inhabitants. These microbial biogeography patterns aren't random. They reflect the underlying physiological and physical properties of each body site. To understand the microbiome, we must first understand this geography, the ways in which different regions of the body create different environments that select for different organisms. Let us begin with your skin, the body's largest organ, a barrier separating your internal world from the external environment. If you could unfold your skin completely and lay it flat, it would cover roughly 2 square meters, about the size of a beach towel. But to call skin a flat surface would be profoundly misleading. At the microscopic scale, skin is a landscape of staggering complexity and variation. Scientists who study the skin microbiome typically divide it into 3 main microenvironments based on physiological properties: sebaceous, moist, and dry. Each presents unique conditions that favor different microbial communities. Sebaceous sites rich in oil-producing sebaceous glands include your face, particularly the T-zone of forehead and nose, your chest, and your upper back. These regions produce sebum, an oily substance composed of triglycerides, wax esters, squalene, and free fatty acids. This creates an environment rich in lipids, which most bacteria cannot metabolize effectively. But certain bacteria have evolved to thrive in these oily conditions. The dominant inhabitant of sebaceous skin is typically Cutibacterium, a genus that was formerly known as Propionibacterium. Cutibacterium acnes, the most famous species in this genus, lives deep in hair follicles and sebaceous glands, feeding on the oils your skin produces. For most people, Cutibacterium species are harmless commensals, breaking down sebum into free fatty acids that actually help maintain skin health by creating a slightly acidic pH that inhibits growth of potential pathogens. But under certain conditions, particularly during adolescence when sebum production increases dramatically, Cutibacterium can contribute to acne when hair follicles become blocked and bacteria proliferate in the trapped oil. Interestingly, when researchers examine microbial diversity across different body sites, sebaceous regions actually show the lowest diversity. The environment is so specialized, so dominated by lipids, that only certain organisms can flourish there. The community is not diverse, but rather consists of high abundances of a few specialized species well adapted to these oily conditions. Moist sites tell a different story entirely. Your armpits, the creases behind your ears, the folds of your groin, between your toes, the inside of your elbows and knees. These warm, humid regions harbor much greater bacterial diversity. Here you find Staphylococcus species, including Staphylococcus epidermidis, which is usually harmless on skin but can cause serious infections if it enters the bloodstream through wounds or medical devices. You find Corynebacterium species in abundance, bacteria that metabolize the compounds in your sweat to produce volatile molecules responsible for body odor. The specific scents produced by your armpit bacteria are deeply personal, a chemical signature as unique to you as your fingerprints. Studies have shown that people can identify others by the smell of their worn t-shirts with surprising accuracy. This isn't magic or supernatural sensitivity. It's simply the fact that each person harbors a slightly different community of Corynebacterium and other bacteria in their armpits, and these different communities produce different mixtures of volatile compounds. Your particular body odor is, quite literally, a message from your microbiome to the world, a chemical communication that operates below the level of conscious awareness, but influences how others perceive you nevertheless. Dry sites, including your forearms, your hands, your legs, present yet another ecological niche. These regions lack the high oil production of sebaceous sites and the moisture of creased areas. They're exposed to more environmental variation, to soap and water and sunlight and air. You might expect them to be microbially impoverished, but the opposite is true. Dry skin sites often show the greatest phylogenetic diversity of any body location, sometimes exceeding even the diversity found in the gut or mouth. These dry areas harbor stable communities of bacteria that were once thought to be mere contaminants from the environment. Gram-negative bacteria, which were assumed to occasionally land on skin from the gut or from environmental sources but not actually live there, are actually permanent residents of dry skin sites, present in substantial abundances. Between your fingers, in those small spaces of the interdigital web, lives a microbial community that changes and fluctuates more over time than communities in more sheltered locations, but nevertheless maintains a characteristic composition distinct from other body sites. Studies that sample many people at many body sites consistently find the same pattern. Where you sample matters more than who you sample. Take 20 people and swab their forearms, and you'll find more similarity among all 20 forearms than between one person's forearm and that same person's armpit. The environment selects the community. Your moist armpit creates conditions, a combination of temperature, humidity, pH, and available nutrients that favor certain bacteria. And while the specific strains might vary from person to person based on genetics, environmental exposures, and chance, the general composition remains remarkably consistent. But this biogeography extends far beyond the skin. Your mouth, perhaps the most ecologically complex environment in your body after your gut, hosts a distinct and incredibly diverse oral microbiome. Your mouth contains multiple sub-environments. Your tongue, with its papillae, creating a textured landscape where anaerobic bacteria can hide from oxygen. Your teeth, providing hard surfaces for biofilm formation. Your gums, where the interface between hard tooth and soft tissue creates unique niches. Your cheeks and palate, softer surfaces with their own characteristic communities. Your saliva, a flowing aqueous environment that carries bacteria throughout the oral cavity, while also providing antimicrobial proteins that limit their growth. In this complex landscape, hundreds of bacterial species coexist. Some you've heard of, like Streptococcus mutans, the primary bacterium responsible for dental cavities. When you eat foods containing sugars, S. mutans and related species ferment these sugars, producing lactic acid as a metabolic byproduct. This acid dissolves the mineral content of your tooth enamel, creating cavities over time if the acid production exceeds your mouth's natural buffering and remineralization capacity. But your oral microbiome contains far more than just the bacteria that cause cavities. It contains Veillonella species, which cannot ferment sugars themselves, but can consume the lactate produced by other bacteria, helping to neutralize acid and protect against cavities. It contains Fusobacterium, bacteria that act as bridges in dental plaque biofilms, binding to many other species and helping to organize the complex three-dimensional structure of plaque. It contains Porphyromonas gingivalis, a bacterium strongly associated with periodontal disease and gum inflammation, which may have connections to cardiovascular disease and other systemic conditions through mechanisms we're only beginning to understand. Your oral microbiome is among the most diverse microbial communities in your body, second only to your gut in terms of bacterial species richness. And it's not just passive. These bacteria interact with your immune system, train your body to distinguish harmless commensals from dangerous pathogens, produce vitamins and metabolites that enter your bloodstream and may influence your risk of diseases far beyond the mouth. Your respiratory system, from nasal passages through sinuses, through trachea and bronchi, and into your lungs, maintains its own microbial ecosystems. For many years, the lungs were thought to be sterile in healthy people, their defenses too formidable for bacteria to colonize. We now know this is wrong. The healthy lung harbors a characteristic microbiome, albeit at much lower densities than the mouth or gut. These respiratory microbes may play important roles in training the local immune system, in resisting colonization by pathogens, and in maintaining respiratory health. Changes in the respiratory microbiome have been associated with conditions like asthma, chronic obstructive pulmonary disease, and cystic fibrosis. Whether these changes cause disease or result from it remains an area of active investigation, But the presence of a lung microbiome in health and its alterations in disease have fundamentally changed how we think about respiratory immunity and pathology. And then there is your gut, the crown jewel of the human microbiome, the most densely populated microbial habitat in your body, perhaps in the entire animal kingdom. If we count microbial abundance across all body sites, roughly 38 trillion of your 39 trillion microbial cells reside in your large intestine. This is where the majority of microbiome research has focused, where the effects on human health are most dramatic and best understood, where the opportunities for therapeutic intervention seem most promising. This is ground zero for understanding how microbes influence human biology. The gut, command center of the microbiome. Imagine your gastrointestinal tract as a journey, a tube roughly 9 meters long running from mouth to anus, interfacing with the external world at both ends. This tube is not just a passive conduit for food. It's one of the body's most important organs, a complex system responsible for breaking down food, absorbing nutrients, defending against pathogens, interacting with trillions of microbes, and communicating with the brain and immune system in ways that influence health throughout the body. The journey begins in your mouth, where mechanical breakdown of food through chewing combines with chemical breakdown through salivary enzymes. Food moves through your esophagus, a muscular tube that pushes each swallow downward through waves of contraction called peristalsis, and enters your stomach. Here, in this acidic cauldron where pH can drop below 2, harsh acids and digestive enzymes, particularly pepsin, begin breaking down proteins. Most microbes cannot survive the stomach's acidic environment. The ones that do, including Helicobacter pylori, which colonizes approximately half the human population worldwide, have evolved remarkable adaptations. H. pylori produces urease, an enzyme that breaks down urea into ammonia, creating a local cloud of alkaline ammonia that neutralizes stomach acid and allows the bacterium to survive. From the stomach, partially digested food enters the small intestine, a winding tube roughly 6 to 7 meters long, divided into 3 sections, the duodenum, jejunum, and ileum. The small intestine is where most nutrient absorption occurs. Its inner surface is covered with millions of finger-like projections called villi, which themselves are covered with even smaller projections called microvilli, creating an enormous surface area estimated at 200 to 300 square meters, roughly the size of a tennis court, all folded and packed into your abdomen. Despite this enormous surface area, the small intestine hosts a relatively modest microbial population compared to what comes next. Digestive enzymes from the pancreas and bile from the liver pour into the duodenum, creating an environment where rapid digestion and absorption leave relatively little for bacteria to feed on. Peristaltic waves move intestinal contents along fairly quickly, giving bacteria limited time to attach and establish stable populations. Still, the small intestine isn't sterile. It harbors around 10,000 to 10 million bacteria per gram of intestinal content, with increasing densities toward the ileum, the final section before the large intestine. Then comes the transition from small intestine to large intestine, marked by the ileocecal valve, a one-way valve that prevents backflow from the colon into the small intestine. Cross this threshold, and you enter a radically different world. The large intestine, also called the colon, roughly 1.5 meters long and significantly wider than the small intestine, is where conditions change dramatically. Here, peristaltic movements slow down substantially. Intestinal contents spend hours in the colon, sometimes days compared to hours in the small intestine. There are no digestive enzymes being secreted, no rapid nutrient absorption occurring. What arrives in the colon is what your body couldn't digest and absorb in the small intestine. primarily complex carbohydrates from plants, fiber, resistant starches, and other indigestible materials, along with dead cells sloughed off from the intestinal lining, bile acids, and various other compounds. And oxygen becomes scarce. The cells lining your colon consume oxygen, creating an anaerobic environment in the lumen where intestinal contents reside. This environment selects strongly for anaerobic bacteria, organisms that don't just tolerate the absence of oxygen, but actually require it, which would be poisoned by exposure to air. These conditions, slow transit, abundant indigestible carbohydrates, anaerobic environment, create perfect conditions for dense bacterial colonization. The concentration of bacteria in the colon reaches 100 billion to 1 trillion cells per gram of intestinal content, among the highest bacterial densities found anywhere in nature. When you pass stool, approximately 30 to 50% of its dry weight consists of bacterial cells and their remains. If you could somehow extract all the different bacterial species from your colon and catalog them, you would find hundreds, or perhaps thousands of distinct species, depending on how finely you want to split them taxonomically. But this diversity isn't evenly distributed across taxonomic groups. Your gut microbiome is dominated by just two bacterial phyla, Firmicutes and Bacteroidetes. Together, these two groups typically account for 70 to 90% of all gut bacteria in healthy Western adults, with smaller contributions from actinobacteria, proteobacteria, and verrucomicrobia. Within Firmicutes, you find families like Lachnospiraceae and Ruminococcaceae, which contain many of the gut's most important butyrate-producing bacteria. Within Bacteroidetes, you find the genus Bacteroides, comprising species like Bacteroides thetaiotiomicron and Bacteroides fragilis, which are champion degraders of complex plant polysaccharides. The specific composition varies from person to person, influenced by genetics, by diet, by medication history, by environmental exposures, by random chance during initial colonization. But despite this person-to-person variation, certain functional capabilities remain relatively consistent. Nearly everyone's gut microbiome can break down common dietary fibers. Nearly everyone's produces short-chain fatty acids. Nearly everyone synthesizes certain vitamins. The specific bacteria performing these functions may differ, but the collective metabolic capabilities of the community remain similar. It's a principle ecologists call functional redundancy, multiple species capable of performing similar ecological roles, which provides resilience and stability to the ecosystem. The molecular conversations, what gut bacteria do for you. The relationship between you and your gut bacteria is not one of host and parasite, not even one of neutral coexistence. It is a mutualism, a partnership in which both parties benefit, evolved over millions of years to be so deeply integrated that neither can thrive optimally without the other. One of the most significant functions your gut microbiome performs is metabolic, breaking down complex carbohydrates that you cannot digest and extracting energy and nutrients that would otherwise be lost. This process, fermentation in the anaerobic environment of your colon, is remarkably sophisticated. It begins with primary degraders, bacteria that possess the enzymes needed to attack specific types of dietary fiber or resistant starch. Different bacteria specialize in different polysaccharides. Some excel at breaking down resistant starch, like that found in whole grains or cooled potatoes. Others target inulin, a fructose polymer found in onions, garlic, and Jerusalem artichokes. Still others specialize in pectins from fruits, beta-glucans from oats, or xylins from plant cell walls. These primary degraders don't consume all the breakdown products themselves. They release smaller oligosaccharides and monosaccharides into the environment, which other bacteria can then scavenge. This creates metabolic webs, networks of bacteria cooperating and competing, sharing and consuming the products of each other's metabolisms. The major end products of this fermentation are short-chain fatty acids, primarily acetate, propionate, and butyrate. These molecules, containing 2, 3, and 4 carbon atoms respectively, are produced in abundance when your diet includes adequate fiber. In a typical Western gut, approximately 500 to 600 millimoles of short-chain fatty acids are produced daily, with typical molar ratios of roughly 60% acetate, 20% propionate, and 20% butyrate. To your gut bacteria, these short-chain fatty acids are waste products, necessary byproducts of fermentation metabolism in an oxygen-free environment. But to you, they are valuable compounds with multiple important functions. Butyrate, in particular, is the preferred energy source for the colonocytes, the epithelial cells lining your colon. These cells absorb butyrate from the gut lumen and transport it inside, where mitochondria oxidize it to generate ATP, the cellular energy currency. Remarkably, butyrate provides an estimated 70 to 90% of the energy needed by colonocytes. Your colon lining is, in a very real sense, living on bacterial waste products. Experiments in germ-free mice, animals raised without any microbiome, have demonstrated how critical this is. Without butyrate-producing bacteria, the colonocytes of germ-free mice show deficits in mitochondrial respiration. They undergo autophagy, a cellular process of self-digestion that occurs during energy starvation. They become dysfunctional. When researchers introduced butyrate-producing bacteria or administered butyrate directly, the colonocytes recovered. Their mitochondrial function was restored. Autophagy ceased. The rescue was due to butyrate acting as an energy source, not primarily through its other signaling functions. But short-chain fatty acids do far more than feed your gut lining. They act as signaling molecules throughout your body, influencing processes far from the gut. After being produced by bacteria, short-chain fatty acids are absorbed by colonocytes, either through passive diffusion or through specific transporters like monocarboxylate transporter 1 and sodium-coupled monocarboxylate transporter 1. Much of the butyrate is consumed locally by colonocytes, but acetate and propionate enter the bloodstream in substantial amounts. Acetate, the most abundant short-chain fatty acid, enters your circulation and travels throughout your body. It can be taken up by muscles, by liver, by brain, by other organs, where it participates in various metabolic processes. It can be incorporated into fatty acid synthesis, into cholesterol synthesis, into the production of acetyl-CoA, a central metabolic molecule. Some acetate even reaches your brain, where it can be used by astrocytes and neurons as an energy source or as a building block for neurotransmitters. Propionate is taken up primarily by your liver, where it can influence glucose production through a process called gluconeogenesis and affect lipid metabolism. Studies have suggested that propionate might improve glucose tolerance and insulin sensitivity, potentially protecting against diabetes, though the mechanisms and optimal dosing remain areas of active research. Beyond their metabolic roles, short-chain fatty acids bind to G-protein-coupled receptors, particularly GPR41 and GPR43, also known as free fatty acid receptors 2 and 3. These receptors are expressed on various cell types throughout your body, including immune cells, enteroendocrine cells in your gut, adipocytes in fat tissue, and cells in your pancreas. When short-chain fatty acids bind to these receptors, they trigger signaling cascades that influence multiple physiological processes. In your gut, short-chain fatty acid signaling promotes the integrity of the intestinal barrier, the critical boundary between the gut contents and your bloodstream. They stimulate the production of mucus, the protective layer that coats the intestinal lining. They enhance the production of antimicrobial peptides that help control bacterial populations and prevent pathogen invasion. They influence the tight junctions between epithelial cells, the protein complexes that seal the gaps and prevent unwanted materials from leaking through. In your immune system, short-chain fatty acids help maintain the balance between inflammation and tolerance. They promote the development of regulatory T cells, crucial immune cells that prevent excessive inflammatory responses and maintain tolerance to harmless antigens. They influence the production of various cytokines, the signaling molecules that coordinate immune responses. They can inhibit histone deacetylases, enzymes that affect gene expression by modifying how DNA is packaged, thereby influencing which genes are turned on or off in immune cells. This immune modulation has far-reaching consequences. Studies have linked short-chain fatty acid production to reduced risk of inflammatory bowel diseases, to protection against allergies and asthma, to proper immune development in infants. The gut microbiome, through its production of short-chain fatty acids and other metabolites, is actively participating in training and regulating your immune system. And the gut microbiome does even more. It synthesizes vitamins that your body cannot produce on its own. Certain gut bacteria produce vitamin K, essential for blood clotting. Others synthesize B vitamins, including thiamine, riboflavin, niacin, pantothenic acid, biotin, folate, and cobalamin. While your diet provides much of these vitamins, your microbiome contributes significantly, and in some cases, like vitamin K production, the microbial contribution may be essential. Gut bacteria metabolize bile acids, the compounds your liver produces to help digest fats. Primary bile acids released into your intestine are transformed by gut bacteria into secondary bile acids through processes like deconjugation and dehydroxylation. These secondary bile acids have different properties than primary bile acids, affecting fat digestion, cholesterol metabolism, and signaling through bile acid receptors throughout your body. The gut microbiome even produces or influences the production of neurotransmitters or their precursors. Specific bacterial species can produce gamma-aminobutyric acid, GABA, the major inhibitory neurotransmitter in your brain. Others produce or influence serotonin production. While most of these neurotransmitters don't cross the blood-brain barrier directly, they can affect the enteric nervous system, that complex network of neurons embedded in your gut wall, sometimes called your second brain because of its size and autonomy. And they can influence signaling through the vagus nerve, the major neural highway between gut and brain, potentially affecting mood, cognition, and behavior. The beginning, how your microbiome forms. Your microbiome's story begins before you're even born. For many years, scientists believed that the womb was a sterile environment, that babies developed in complete isolation from microbial life, encountering microbes for the first time as they passed through the birth canal. This view has been challenged. Bacterial DNA has been detected in amniotic fluid, in placental tissue, in meconium, the first stool passed by newborns. While the significance and viability of these bacteria remains debated, with some researchers suggesting the DNA represents dead cells or contamination rather than living communities, the evidence increasingly suggests that microbial exposure, or at least exposure to microbial components, begins before birth. During pregnancy, the maternal microbiome undergoes changes. The vaginal microbiome, in particular, shifts in composition, typically becoming enriched in lactobacillus species, which dominate in most pregnant women. These changes may prepare the maternal microbiome for its critical role in seeding the infant's first microbial communities. Then comes birth, the most dramatic microbial colonization event in human life. The specific route of birth, vaginal delivery versus cesarean section, has profound effects on which microbes initially colonize the newborn. During vaginal delivery, the baby passes through the birth canal, a journey that coats the infant's skin, mouth, and airways with bacteria from the mother's vagina. These bacteria, predominantly Lactobacillus species, in healthy mothers, along with some Prevotella, Snethia, and other vaginal inhabitants, become the infant's first microbial colonizers. The baby swallows these bacteria, establishing initial populations in the gut. The baby breathes them in, seeding the respiratory tract. The baby's skin is covered with them, initiating colonization of skin sites. This vaginal microbiome transfer is not random. It appears to be an evolutionarily conserved process, a feature of mammalian reproduction that serves important functions. The bacteria that colonize infants during vaginal birth are typically non-pathogenic, adapted to the human body, and compatible with infant immune development. They set the stage for subsequent microbial succession, the ordered sequence of community changes that will occur over the coming months and years. Babies born by cesarean section miss this microbial transfer. They don't pass through the birth canal. Instead, they're lifted directly from the uterus through a surgical incision in the mother's abdomen. Their first microbial exposures come not from the vagina, but from the mother's skin, from the hands of medical personnel, from the hospital environment. The initial colonization is dramatically different. Studies comparing vaginally delivered and cesarean-born infants consistently find significant differences in early microbiome composition. Cesarean-born babies show reduced colonization by Lactobacillus and Bacteroid species. They show increased colonization by skin-associated bacteria like Staphylococcus and environmental bacteria like Acinetobacter. Their gut microbiomes take longer to diversify, longer to accumulate beneficial species like Bifidobacterium that should predominate during breastfeeding. These differences can persist for months or even years. Some studies have found microbiome differences between cesarean and vaginally born children detectable at 7 years of age. The clinical significance of these differences remains an area of active investigation, but epidemiological studies have found associations between cesarean birth and increased risk of various health conditions later in life, including obesity, asthma, allergies, type 1 diabetes, and celiac disease. Recognizing these effects, some researchers have investigated interventions to restore the vaginal microbiome transfer in cesarean-born infants. The procedure, sometimes called vaginal seeding or microbial bathing, involves swabbing the newborn's mouth, face, and body with gauze that has been incubated in the mother's vagina just before the cesarean section. Early studies showed that this could partially restore the microbiome, making cesarean-born infants' microbial profiles more similar to vaginally delivered infants, at least in the first weeks of life. However, vaginal seeding remains controversial. Medical societies have raised concerns about safety, noting that the procedure could potentially transfer harmful bacteria or viruses from mother to infant, including Group B Streptococcus, herpes simplex virus, or other pathogens. Mothers need to be carefully screened before the procedure. Long-term effects on health outcomes remain unknown. While the intervention can alter microbiome composition in the short term, whether this translates to meaningful health benefits requires much more research. In the hours and days after birth, regardless of delivery mode, the infant's microbiome begins a remarkable journey of development and maturation. This process is influenced by countless factors, creating a unique microbial fingerprint for each individual. Feeding method plays a particularly crucial role. Breast milk is not just nutrition for the infant. It's a complex biological fluid designed to nourish beneficial bacteria. Human milk oligosaccharides, complex sugars that make up the third most abundant component of breast milk after lactose and lipids, cannot be digested by the infant. They pass through the stomach and small intestine intact. They're not there to feed the baby, they're there to feed the baby's bacteria. Bifidobacterium species, particularly Bifidobacterium longum subspecies infantis, have evolved specialized gene clusters that allow them to consume human milk oligosaccharides efficiently. These bacteria dominate the gut of exclusively breastfed infants, often comprising 50 to 90% of the total gut microbiome. They create an acidic environment by producing lactate and acetate, which helps inhibit growth of potential pathogens. They stimulate proper immune development. They establish themselves as the dominant colonizers during this critical developmental window. Breast milk provides more than just oligosaccharides to feed beneficial bacteria. It contains living bacteria itself, a breast milk microbiome that includes Staphylococcus, Streptococcus, and Lactobacillus species. It delivers immunoglobulin A, antibodies that coat bacteria and prevent them from attaching to the intestinal wall, helping to regulate which bacteria can colonize. It provides cytokines and other immune factors that help train the infant's developing immune system to tolerate gut bacteria rather than mounting inflammatory attacks against them. Formula-fed infants show different microbiome development. Without human milk oligosaccharides, their guts are not as strongly selected for bifidobacterium. They show greater microbial diversity earlier, with increased abundances of bacteria from families like Enterobacteriaceae, Clostridiaceae, and Lachnospiraceae. This isn't necessarily harmful. Most formula-fed babies develop healthy microbiomes and thrive, but it represents a different developmental trajectory with potentially different implications for immune education and disease risk. As the infant grows, as solid foods are introduced around 4 to 6 months of age, the gut microbiome undergoes dramatic shifts. The introduction of complex carbohydrates from fruits, vegetables, and grains provides new substrates for bacterial fermentation. Firmicute species, particularly members of families like Lachnospiraceae and Ruminococcaceae, increase in abundance as they begin breaking down these more complex dietary components. The microbiome becomes more diverse, more similar to the adult pattern. By around 3 to 5 years of age, the child's gut microbiome has largely stabilized into an adult-like configuration in terms of overall structure and dominant bacterial groups. Those specific strains and relative abundances will continue to shift throughout life in response to diet, medications, illness, and environmental changes. This early life period, particularly the first 1,000 days from conception through the child's second birthday, represents a critical window for microbiome establishment. Disruptions during this window can have lasting effects. Antibiotics given to infants or young children can profoundly alter the developing microbiome, depleting beneficial species and allowing potential pathogens to proliferate. Studies have found associations between early life antibiotic exposure and increased risk of obesity, allergies, asthma, and other conditions later in childhood, likely mediated through effects on microbiome development and immune education. The developing microbiome is not just acquiring bacteria, it's establishing a relationship with the developing immune system. Throughout infancy and early childhood, immune cells are learning to distinguish self from non-self, harmless from harmful, commensal from pathogen. The presence of a diverse, healthy microbiome provides essential education signals for this process. Germ-free mice, animals raised from birth without any microbiome, show profound immune deficiencies. Their gut-associated lymphoid tissues are severely underdeveloped. They have fewer and smaller Peyer's patches, the specialized immune organs in the small intestine. They have fewer immunoglobulin-producing cells. They cannot mount effective immune responses to pathogens. Even introducing normal microbes later in life cannot fully reverse these deficits. Some aspects of immune education appear to have critical developmental windows that, once passed, cannot be reopened. In humans, we cannot perform such experiments, but observational studies suggest similar principles apply. Children raised in overly sterile environments with limited microbial exposure show increased rates of allergies and asthma. Children who grow up on farms exposed to a greater diversity of environmental microbes through contact with animals and soil show reduced rates of allergic diseases. The hygiene hypothesis, now refined into concepts like the old friends hypothesis, suggests that reduced microbial exposure in modern industrialized societies may contribute to rising rates of allergic and autoimmune diseases by depriving the developing immune system of essential educational signals. The gut-brain axis, microbes that influence mind. For most of medical history, the brain was considered privileged territory, isolated from the body's messier biological processes by the blood-brain barrier, that highly selective filter that prevents most substances in the bloodstream from entering brain tissue. The gut and the brain seem to occupy separate realms. One dealt with crude necessities like digesting food. The other handled refined complexities like thought, emotion, consciousness. They might be connected through neural and hormonal pathways, but they fundamentally operated independently. This view has collapsed. In its place has emerged an understanding of the gut-brain axis, a bidirectional communication highway linking your gastrointestinal system to your central nervous system through multiple pathways: neural, immune, endocrine, and metabolic. And the microbiome, it turns out, is a major participant in these conversations, influencing brain function and behavior in ways we're only beginning to understand. The most direct communication pathway is the vagus nerve, the 10th cranial nerve, the longest and most complex of the cranial nerves. The vagus nerve extends from the brainstem, descending through the neck and chest and into the abdomen, sending branches to the heart, lungs, stomach, liver, pancreas, and intestines. About 80 to 90% of the vagus nerve fibers are sensory, carrying information from organs to the brain rather than commands from brain to organs. The vagus nerve is constantly monitoring the state of your internal organs and reporting back to your brain. In your gut, vagal nerve endings detect various signals: mechanical stretch indicating fullness, chemical signals including hormones released by enteroendocrine cells, immune signals including cytokines, and potentially direct signals from bacteria or bacterial metabolites. These sensory neurons project to the nucleus tractus solitarius in your brainstem, which then relays information to multiple brain regions involved in regulating appetite, mood, stress response, and other functions. Studies in mice have shown that severing the vagus nerve blocks certain microbiome effects on brain and behavior. For example, the behavioral effects of probiotic bacteria, including reduced anxiety-like behavior and altered stress responses, are abolished in vagotomized animals. This demonstrates that at least some microbiome brain communication requires an intact vagus nerve. But the vagus nerve is just one pathway. The microbiome influences the brain through multiple routes simultaneously. Consider neurotransmitters. Your gut microbiome produces or influences the production of virtually every neurotransmitter known to neuroscience. Certain bacteria can synthesize gamma-aminobutyric acid, GABA, the major inhibitory neurotransmitter in your brain. Others produce or influence the production of serotonin, often called the happiness molecule, which regulates mood, appetite, sleep, and other functions. About 90 to 95% of your body's serotonin is actually produced in your gut, not your brain, by enterochrome affin cells in the intestinal lining, a process influenced by gut bacteria. Bacteria can produce dopamine, the neurotransmitter associated with reward and motivation. They can produce norepinephrine, involved in arousal and stress responses. They can produce acetylcholine, crucial for memory and muscle control. They produce or influence histamine, involved in inflammation and arousal. The gut is, in a very literal sense, a neurotransmitter factory, with bacteria playing crucial manufacturing roles. Now, most of these gut-produced neurotransmitters cannot cross the blood-brain barrier directly. They don't enter the brain to bind to neurons, but they don't need to. They can affect the enteric nervous system, that extensive network of hundreds of millions of neurons embedded in your gut wall. They can influence vagal signaling. They can affect gut motility, gut sensation, gut hormone release. And through these indirect routes, they can influence brain function and behavior. The microbiome also communicates with the brain through the immune system. Your gut contains more immune cells than any other organ in your body. These immune cells are in constant dialogue with gut bacteria, sampling bacterial components, releasing cytokines and other signaling molecules in response. Many of these cytokines can cross the blood-brain barrier or signal through other routes to affect brain function. During infection or inflammation, when large amounts of pro-inflammatory cytokines are released, people experience what's called sickness behavior, fatigue, reduced appetite, social withdrawal, difficulty concentrating, low mood. These are not side effects but coordinated responses, orchestrated by the immune system to help fight infection. The cytokines are signaling to the brain that the body is fighting off a threat, and the brain responds by shifting priorities away from social interaction and cognitive work toward rest and recovery. Chronic low-grade inflammation, which can result from gut dysbiosis, may produce subtler but persistent effects on mood and cognition through similar mechanisms. Studies have found elevated levels of inflammatory markers in people with depression, and some researchers have proposed that depression, at least in some cases, may be an inflammatory condition linked to immune dysfunction that originates in or is influenced by the gut. The hypothalamic pituitary adrenal axis, your body's main stress response system, is also influenced by the gut microbiome. When you encounter a stressor, your hypothalamus releases corticotropin-releasing hormone, which stimulates your pituitary gland to release adrenocorticotropic hormone, which stimulates your adrenal glands to release cortisol and other stress hormones. These hormones help you respond to threats by mobilizing energy, increasing alertness, and temporarily suppressing non-essential functions like digestion and reproduction. Germ-free mice show exaggerated stress responses. When exposed to stressors, they show higher and more prolonged elevation of stress hormones compared to normal mice with intact microbiomes. Colonizing germ-free mice with normal bacteria can partially normalize this response, particularly if done early in life. This suggests the microbiome helps calibrate the stress response system during development. And then there are the metabolites, particularly the short-chain fatty acids we discussed earlier. These bacterial fermentation products can cross the blood-brain barrier and directly influence brain function. They affect the integrity of the blood-brain barrier itself, potentially influencing what can or cannot enter the brain. They influence the activity of microglia, the brain's resident immune cells, which play crucial roles in maintaining neural connections and responding to injury or infection. Short-chain fatty acids can inhibit histone deacetylases in brain cells, affecting gene expression through epigenetic mechanisms. They can influence the production of brain-derived neurotrophic factor, a protein crucial for neuronal survival, growth, and plasticity. They participate in the synthesis of neurotransmitters. Through these multiple mechanisms, bacterial metabolites can influence brain chemistry, neural function, and ultimately behavior. All of these pathways, neural, immune, endocrine, and metabolic, work together, creating a complex web of bidirectional communication between gut and brain. And increasingly, research suggests that disruptions in this gut-brain axis may contribute to neurological and psychiatric conditions. Depression and anxiety, two of the most common mental health disorders worldwide, affecting hundreds of millions of people, have been linked to alterations in the gut microbiome. Multiple studies have found differences in gut microbial composition between people with depression and healthy controls. The specific differences vary between studies, likely reflecting the heterogeneity of depression and the many factors that influence the microbiome, but patterns are emerging. Depressed individuals often show reduced microbial diversity and altered abundances of specific bacterial groups. More compellingly, some studies suggest these microbial differences may be causal, not just correlative. In one striking experiment, researchers collected stool samples from people with major depressive disorder and from healthy controls. They transplanted these stool microbiomes into germ-free rats. Remarkably, rats that received microbiomes from depressed humans began showing depressive-like behaviors, including anhedonia, loss of interest in rewards, and behavioral despair. Rats that received microbiomes from healthy humans did not show these behaviors. This suggests that the microbiome can transmit behavioral phenotypes, that something in the depressed microbiome is contributing to depressive symptoms. What that something might be remains unclear. It could be reduced production of beneficial metabolites, like short-chain fatty acids or tryptophan metabolites. It could be increased production of harmful metabolites. It could be altered neurotransmitter production. It could be effects on inflammation and immune function. Most likely, it's some complex combination of multiple mechanisms operating simultaneously. Autism spectrum disorder has also been associated with distinct gut microbiome profiles and frequent gastrointestinal symptoms. Children with autism often show reduced microbial diversity, altered abundances of specific bacterial groups, and increased intestinal permeability compared to neurotypical children. Whether these microbiome differences contribute to autism's core symptoms, exacerbate them, or result from them remains hotly debated. Small studies have explored whether interventions targeting the microbiome, including strict dietary interventions and even fecal microbiota transplantation, might improve behavioral symptoms, with some promising preliminary results, but also many unanswered questions about mechanisms, safety, and efficacy. Parkinson's disease, the second most common neurodegenerative disorder after Alzheimer's, characterized by death of dopamine-producing neurons in the brain, may actually begin in the gut decades before motor symptoms appear. The hallmark pathology of Parkinson's involves abnormal aggregates of a protein called alpha-synuclein forming inside neurons. Evidence suggests these aggregates can form first in the enteric nervous system and then travel via the vagus nerve to the brain. In mice, injecting alpha-synuclein aggregates into the gut wall leads to Parkinson's-like pathology that spreads to the brain in a time-dependent manner. Cutting the vagus nerve prevents this spread. In humans, studies have found that people who underwent full vagotomy, surgical severing of the vagus nerve, decades earlier for treatment of ulcers, show reduced risk of Parkinson's disease, suggesting that the vagus nerve may be a route by which the disease spreads from gut to brain. Gut dysbiosis has been documented in Parkinson's patients, with specific alterations in bacterial composition and reduced production of short-chain fatty acids. Whether these microbiome changes contribute to disease initiation or progression remains an area of intense investigation, but the gut-first theory of Parkinson's disease is gaining support from multiple lines of evidence. Alzheimer's disease, multiple sclerosis, epilepsy, and other neurological conditions are all being investigated for potential microbiome connections. The field is still young. Many claims remain speculative, but the underlying principle is becoming increasingly clear. Your gut microbiome influences your brain, and your brain influences your gut microbiome in a complex feedback loop that affects cognition, emotion, behavior, and neurological health. The dark side, when balance breaks, for all the beneficial functions the microbiome performs, all the vital roles it plays in digestion, metabolism, immune function, and health maintenance, it can also become a source of disease. Dysbiosis, the disruption of normal microbial balance, has been implicated in a growing and somewhat alarming list of health conditions, from inflammatory bowel diseases to metabolic disorders to cancer to cardiovascular disease. Inflammatory bowel diseases, primarily Crohn's disease and ulcerative colitis, involve chronic inflammation of the digestive tract. The exact causes remain unclear, involving complex interactions between genetics, immune dysfunction, environmental factors, and the microbiome. But evidence strongly implicates gut dysbiosis in disease pathogenesis. Patients with inflammatory bowel disease show markedly reduced microbial diversity compared to healthy individuals. They show depletion of certain beneficial bacteria, particularly firmicute species from families like Lactospiraceae and Ruminococcaceae, which produce anti-inflammatory short-chain fatty acids. They show increased abundances of potentially harmful bacteria, including adherent invasive E. coli strains that can penetrate the intestinal lining, and Fusobacterium species associated with inflammation. The mucus layer protecting the intestinal wall is often thinner and more penetrable in inflammatory bowel disease patients. Bacteria that normally reside at a safe distance in the outer mucus layer or in the gut lumen can reach and adhere to the epithelium, triggering immune responses. The production of antimicrobial peptides by gut epithelial cells may be reduced, further compromising barrier function. In this inflamed environment, bacteria that would normally be harmless commensals can become inflammatory agents, not because they've changed, but because the immune system's tolerance has broken down. Whether dysbiosis causes inflammatory bowel disease or results from the inflammation remains debated. Most likely, the relationship is bidirectional and self-reinforcing. Genetic susceptibility and environmental triggers may initiate low-grade inflammation and subtle immune dysfunction. This creates conditions favoring dysbiosis. The dysbiosis amplifies inflammation. The inflammation further disrupts the microbiome. A vicious cycle develops that eventually manifests as clinical disease. Medications taken to treat the disease, particularly antibiotics and immunosuppressants, can further alter the microbiome, adding another layer of complexity. Obesity and metabolic syndrome represent another area where microbiome alterations have been consistently documented. The gut microbiomes of obese individuals differ from those of lean individuals in ways that have been replicated across multiple studies, though the specific details vary. Early studies suggested that obese people have altered ratios of major bacterial phyla, with increased firmicutes and decreased bacteroidetes, though subsequent research has shown the picture is more complex. What seems clearer is that the gut microbiome influences energy harvest from food, fat storage, insulin sensitivity, and inflammation, all factors relevant to obesity and metabolic health. Some gut microbial communities may be more efficient at extracting calories from food, breaking down otherwise indigestible carbohydrates, and making the energy available for absorption. The energy harvesting hypothesis suggests that certain microbiomes contribute extra calories through this enhanced fermentation, potentially promoting weight gain. Other mechanisms may involve inflammation. Obesity is associated with chronic low-grade inflammation, and gut dysbiosis may contribute to this. When the intestinal barrier becomes more permeable, bacterial components like lipopolysaccharide can leak into the bloodstream, triggering inflammatory responses. This metabolic endotoxemia has been linked to insulin resistance, fatty liver disease, and other metabolic complications. Short-chain fatty acid production is altered in obesity, with some studies showing reduced butyrate production. Given butyrate's anti-inflammatory properties and its effects on metabolism and satiety, this reduction could contribute to metabolic dysfunction and difficulty regulating food intake. Fascinatingly, microbiome transplantation experiments suggest the microbiome can transmit metabolic phenotypes. When researchers transplant gut microbiomes from obese mice into germ-free mice, the recipients gain more fat than mice receiving microbiomes from lean donors, even when both groups eat the same diet. Similar results have been found using human microbiomes. These experiments strongly suggest that the microbiome plays a causal role in obesity, not just a correlative one. Type 2 diabetes, characterized by insulin resistance and impaired glucose regulation, shows similar microbiome associations. Diabetic patients show altered gut microbial composition and reduced production of short-chain fatty acids, particularly butyrate and propionate. These fatty acids influence glucose homeostasis through multiple mechanisms, including effects on insulin secretion, glucose production by the liver, and insulin sensitivity in peripheral tissues. Colorectal cancer, one of the most common cancers worldwide, has been linked to specific changes in the gut microbiome. Certain bacteria, including Fusobacterium nucleatum and toxin-producing strains of Bacteroides fragilis, are enriched in colorectal tumors. These bacteria may promote cancer development through multiple mechanisms, producing compounds that damage DNA, inducing chronic inflammation, interfering with tumor suppressor pathways, or promoting an immunosuppressive environment that allows tumors to evade immune detection. The microbiome also influences how patients respond to cancer treatments. Some bacteria enhance the effectiveness of certain chemotherapy drugs, while others reduce it. The microbiome influences responses to immunotherapy, the revolutionary cancer treatment that unleashes the immune system to attack tumors. Patients with more diverse gut microbiomes and higher abundances of specific beneficial bacteria show better responses to immune checkpoint inhibitors. This has led to trials testing whether manipulating the microbiome through diet, probiotics, or even fecal microbiota transplantation can improve cancer treatment outcomes. Cardiovascular disease, the leading cause of death globally, may also have microbiome connections. Certain gut bacteria metabolize dietary compounds like choline and carnitine, found in red meat, eggs, and other animal products, into trimethylamine. This molecule is absorbed and carried to the liver, where it's oxidized to trimethylamine N-oxide, TMAO. Elevated TMAO levels have been associated with increased risk of heart attack and stroke in multiple studies, possibly through effects on cholesterol metabolism, promoting foam cell formation in arteries, and enhancing platelet reactivity. Allergies, asthma, eczema, conditions involving inappropriate immune responses have been linked to reduced microbial diversity and specific alterations in the gut microbiome, particularly in early life. The hygiene hypothesis and its descendants suggests that lack of diverse microbial exposure during critical developmental periods leads to skewed immune development, with the immune system becoming hyperreactive to harmless substances. Even conditions not obviously related to the gut show microbiome associations. Chronic kidney disease, liver disease, rheumatoid arthritis, multiple sclerosis, and various other autoimmune conditions have all been linked to altered gut microbial profiles. The mechanisms likely involve systemic inflammation, altered metabolite production, bacterial translocation, and immune dysfunction originating from or influenced by gut dysbiosis. But establishing causation in all these conditions remains challenging. Correlation is not causation. Does dysbiosis cause disease, or does disease cause dysbiosis? In many cases, the relationship is likely bidirectional, cyclical, and self-reinforcing. A genetically susceptible individual may develop subtle microbiome alterations in response to environmental exposures or dietary patterns. These alterations trigger mild inflammation or metabolic changes. The inflammation further disrupts the microbiome. Medications taken for early symptoms alter the microbiome further. The disease process itself, through effects on diet, motility, immune function, creates more dysbiosis. Eventually, this tangled web of cause and effect manifests as clinical disease. Disentangling these relationships requires careful longitudinal studies following people over time, intervention studies testing whether manipulating the microbiome prevents or treats disease, and mechanistic studies in animal models where causation can be demonstrated more definitively. This work is ongoing, and our understanding grows more nuanced and sophisticated each year. But one thing is clear: the microbiome is not just a bystander in disease, but an active participant whose manipulation may offer therapeutic opportunities. The antibiotic paradox. Antibiotics rank among the greatest medical achievements in human history. Since penicillin was mass-produced during World War II, antibiotics have saved countless lives, transformed once deadly infections into minor inconveniences, and enabled medical procedures from surgery to organ transplantation to cancer chemotherapy that would be impossibly dangerous without reliable infection control. The average life expectancy in developed countries increased dramatically in the mid-20th century, and antibiotics deserve substantial credit for this achievement. But antibiotics come with a cost that we're only now fully appreciating. That cost is measured in damage to the microbiome, collateral destruction of beneficial bacteria, alongside the pathogens being targeted. And this damage, accumulated over decades of antibiotic use in individuals and populations, may contribute to some of the chronic diseases plaguing modern society. By design, antibiotics kill or inhibit bacteria. That's their purpose, their virtue, their life-saving function. But most antibiotics, particularly the broad-spectrum varieties that are most commonly prescribed, cannot discriminate between pathogenic bacteria causing infection and beneficial commensal bacteria maintaining gut health. When you take a course of antibiotics for a respiratory infection caused by bacteria in your lungs, the antibiotics enter your bloodstream, circulate throughout your body, and kill bacteria everywhere they can reach. The bacteria in your gut, which had nothing to do with your illness, die as collateral damage. The effects can be dramatic and immediate. Within days of starting antibiotics, the gut microbiome undergoes profound changes. Microbial diversity plummets as antibiotic-sensitive bacteria are killed off. Bacterial groups that normally dominate may be reduced to barely detectable levels. The metabolic output of the microbiome shifts dramatically, with sharp reductions in short-chain fatty acid production and other beneficial metabolites. The ecological niches left empty by dead bacteria may be rapidly colonized by antibiotic-resistant bacteria or opportunistic pathogens that had previously been suppressed by competition from the normal microbiota. Clostridioides difficile infection exemplifies this dynamic perfectly. C. difficile is a spore-forming bacterium that's present in small numbers in many people's intestines without causing problems, kept in check by the dense populations of other bacteria that outcompete it for resources and produce inhibitory compounds. But antibiotic treatment, especially with broad-spectrum agents like clindamycin, fluoroquinolones, or third-generation cephalosporins, can decimate the normal microbiota. With competition removed, C. difficile spores germinate, populations explode, and the bacteria produce toxins that cause severe, sometimes life-threatening diarrhea. C. difficile infection is almost entirely a disease of people who've recently received antibiotics, a perfect example of how disrupting the microbiome can enable pathogens to invade. Some of the antibiotic-induced changes are temporary. After antibiotic treatment ends, many people's microbiomes begin recovering. Bacterial populations rebound, diversity increases again, metabolic functions are restored, but recovery is often incomplete. Multiple studies have found that even a single course of antibiotics can produce persistent changes in gut microbial composition detectable months or even years later. Certain bacterial strains that were present before antibiotics may not return. Others may never reach their previous abundances. The community that reforms is not identical to what existed before. Multiple courses of antibiotics, common in modern medicine, can produce cumulative effects. Each course causes disruption. Each recovery is incomplete. Over years or decades, the microbiome of someone who's received many antibiotic courses may drift substantially from what it would have been without such exposures. We don't yet know the long-term health implications of this drift, but there are reasons for concern. The consequences of antibiotic-induced dysbiosis extend beyond the gut. Reduced microbial diversity is associated with increased susceptibility to various infections, not just C. difficile, but also resistant urinary tract infections, pneumonia, and sepsis. The normal microbiota provides what ecologists call colonization resistance, using up resources, producing inhibitory compounds, and occupying ecological niches in ways that prevent pathogens from gaining a foothold. Strip away this protective community with antibiotics, and pathogens have opportunities to invade. Antibiotics in early life are particularly concerning because they disrupt microbiome development during a critical window. Infants and young children receive antibiotics frequently, often for ear infections, respiratory infections, urinary tract infections, and other common childhood ailments. Some of these antibiotic prescriptions are medically necessary and potentially life-saving, but others may be prescribed inappropriately for viral infections that won't respond to antibiotics or as precautionary measures when infection isn't clearly established. The first 1,000 days of life, from conception through the child's second birthday, represent a critical period for microbiome establishment and immune education. Disrupting this process with antibiotics can have lasting effects. Large epidemiological studies involving hundreds of thousands of children have found associations between early life, antibiotic exposure, and increased risk of obesity, type 1 diabetes, allergies, asthma, eczema, inflammatory bowel disease, and celiac disease later in childhood and adolescence. The mechanisms likely involve disrupted immune development due to altered microbial exposures during critical windows, metabolic reprogramming due to changed bacterial metabolite production, and persistent alterations in gut microbial composition. A Danish study following over 700,000 children found that antibiotic exposure in the first year of life was associated with increased risk of asthma, with risk increasing with each additional antibiotic course. A British study of nearly 800,000 children found similar associations between early antibiotics and allergies. A Finnish study found increased risk of type 1 diabetes associated with antibiotic exposures in infancy. These are observational studies, not randomized trials, so they cannot definitively prove causation. Children who receive more antibiotics may differ from those who don't in ways that independently affect disease risk. They may have more infections because of underlying immune vulnerabilities. They may have different environmental exposures, different diets, different genetics. Teasing apart these confounding factors is challenging. But animal studies where causation can be demonstrated more definitively support the plausibility of antibiotic effects. In mice, antibiotic treatment early in life causes lasting changes in the microbiome, metabolic alterations including increased adiposity and altered glucose metabolism and skewed immune development with increased susceptibility to allergic and inflammatory conditions. Transferring microbiomes from antibiotic-treated mice to germ-free mice transmits some of these phenotypes, demonstrating that microbiome alterations are causally involved. This is not an argument against antibiotics when they're truly needed. When a child has bacterial meningitis or pneumonia, when sepsis threatens life, antibiotics are essential and life-saving. The problem is overuse, inappropriate use, and unnecessary use. Antibiotics prescribed for viral upper respiratory infections, which they cannot treat. Antibiotics given prophylactically when infection risk is low. Antibiotics continued for longer than necessary. Each unnecessary antibiotic course carries risks without offsetting benefits. The gut microbiome has also become a reservoir for antibiotic resistance genes, adding another dimension to the problem. Bacteria can share genes horizontally, passing genetic material including resistance genes from one species to another through plasmids, transposons, and other mobile genetic elements. When you take antibiotics, you create strong selection pressure for resistance. Bacteria carrying resistance genes survive and multiply while sensitive bacteria die. The resistance genes can spread through the microbial community, even to species that weren't present when the antibiotic was taken. Studies sequencing the metagenomes of human gut microbiomes have found hundreds of different antibiotic resistance genes. Some of these confer resistance to antibiotics still in common use. Others confer resistance to multiple drug classes simultaneously. This vast reservoir of resistance genes in the collective human microbiome represents a significant public health threat, as these genes can potentially transfer to pathogenic bacteria, making infections harder or impossible to treat. Researchers are exploring strategies to mitigate antibiotic damage to the microbiome. Probiotics, live beneficial bacteria consumed during or after antibiotic treatment, have been proposed as a way to help restore the microbiome. Some studies show benefits, particularly for preventing C. difficile infection in people taking antibiotics, but results have been mixed. For other outcomes, and some evidence suggests that probiotics might actually delay natural microbiome recovery by occupying ecological niches that native bacteria need to recolonize. More promising may be autologous fecal microbiota transplantation, storing a person's own stool before planned antibiotic treatment and then reintroducing it afterward. A small study showed this could rapidly restore the pre-antibiotic microbiome within days, whereas spontaneous recovery took months and was incomplete. But practical challenges, regulatory hurdles, and the logistics of stool banking limit widespread adoption of this approach. Another strategy involves enzymatic degradation of antibiotics in the gut. An investigational drug called ribaxamase, a beta-lactamase enzyme administered orally during intravenous antibiotic treatment, can break down certain antibiotics in the intestine before they damage the gut microbiome. potentially preserving gut bacteria while still allowing the antibiotic to work systemically. Early trials showed this reduced antibiotic-induced dysbiosis and decreased C. difficile colonization, though the drug remains experimental. The antibiotic paradox, their necessity for treating life-threatening infections weighed against their collateral damage to beneficial microbes, exemplifies a broader challenge in medicine: how to target disease precisely without harming essential biological systems. As our understanding of the microbiome grows, so does the imperative to use antibiotics more judiciously, preserving this powerful tool while protecting the microbial communities on which our health depends. Feeding the invisible ecosystem, diet, and the microbiome. If your gut microbiome is an ecosystem, then food is its weather and climate, the driving environmental force that determines which species thrive and which struggle to survive. What you eat doesn't just feed your human cells. It profoundly shapes the trillions of microbes in your gut, and they respond to dietary changes with remarkable speed and sensitivity. The composition of your microbiome can shift detectably within 24 hours of changing your diet, and more dramatically over days and weeks. The most important dietary factor for the gut microbiome is fiber, those complex carbohydrates found in plants that your human digestive enzymes cannot break down. While fiber is often treated as a single entity, it actually encompasses a remarkably diverse array of chemical structures. Cellulose, the structural component of plant cell walls, is a polymer of glucose molecules linked by beta bonds that human enzymes cannot cleave. Hemicelluloses include diverse polysaccharides like xylins, mannans, and glucans, with branched structures. Pectins, found abundantly in fruits, consist of galacturonic acid chains with side branches of various sugars. Resistant starches, despite being composed of glucose like regular starch, resist digestion due to their physical structure. Inulins and fructans are polymers of fructose. Beta-glucans, found in oats and barley, have specific structural features that affect their fermentation properties. All of these fibers pass through your stomach and small intestine largely intact, arriving in your colon where resident bacteria possess the enzymes needed to break them down. But different bacteria specialize in different fibers, creating a principle called selective fermentation. Bifidobacterium species excel at fermenting fructans and galacto-oligosaccharides. Bacteroides thetioteiomicron can degrade an astonishing variety of plant polysaccharides thanks to its extensive enzymatic toolkit. Ruminococcus bromii specializes in resistant starch, possessing unique enzymes that can penetrate starch granules and begin breaking them down from the inside. Members of the Lactospiraceae family produce much of the butyrate in your gut, using fermentation products from other bacteria as their starting materials. This metabolic specialization means that dietary diversity matters tremendously. A diet containing many types of plant-based foods, providing many types of fiber, supports a diverse microbiome with many species occupying different metabolic niches. Each bacterial species finds its preferred substrate. Competition and cooperation create complex food webs, where the waste products of one species become the nutrients for another. Conversely, a low-fiber diet, typical of Western industrialized countries where average fiber intake is often below 20 grams per day, half the recommended amount or less, starves beneficial fiber-fermenting bacteria. Studies comparing populations on high-fiber traditional diets to those on low-fiber Western diets consistently find dramatic differences in microbiome composition. People consuming high-fiber diets show greater microbial diversity, higher abundances of beneficial bacteria from families like Lactospiraceae and Ruminococcaceae. and higher production of protective short-chain fatty acids. When deprived of dietary fiber, gut bacteria don't simply die. They adapt, finding alternative food sources. Some bacteria possess enzymes capable of degrading the mucus layer that protects your intestinal lining. Under normal circumstances with adequate fiber intake, these mucus-degrading bacteria are a minor part of the community, their enzymatic capabilities held in check by abundant alternative food sources. But fiber deprivation changes the equation. Studies in mice fed fiber-free diets have shown that certain bacterial species upregulate expression of mucus-degrading enzymes and begin consuming the protective mucus barrier itself. This has alarming implications. The mucus layer is your gut's first line of defense, a physical barrier separating bacteria from the epithelial cells that line your intestine. Thinning of this barrier increases gut permeability, potentially allowing bacterial products to reach the epithelium and trigger inflammation. This mechanism may link low-fiber diets to inflammatory bowel disease, metabolic dysfunction, and other conditions associated with increased intestinal permeability. The amount of fiber matters too, as demonstrated by intervention studies. When researchers take people eating low-fiber diets and supplement them with specific types of fiber, the microbiome responds within days. Resistant starch supplementation increases abundances of Ruminococcus bromide and related species within a week. Inulin supplementation boosts bifidobacterium populations. These compositional changes correlate with increased short-chain fatty acid production measurable in stool samples. But not everyone responds to fiber supplementation the same way. Your microbiome's baseline composition determines how effectively it can extract nutrients and produce metabolites from any given fiber source. If you lack Ruminococcus bromide, the keystone species for resistant starch degradation, eating more resistant starch may not substantially increase butyrate production in your gut. Other bacteria can eventually adapt to fill this ecological role, but the process takes time and may be inefficient. This person-to-person variation, rooted in differences in baseline microbiome composition, explains why dietary interventions and probiotic supplements show such variable effects across individuals. Protein and fat in the diet also profoundly influence the microbiome, though their effects are more complex and context-dependent than fiber. High-protein diets, particularly those rich in animal protein from red meat and processed meats, increase protein fermentation in the distal colon, where carbohydrates have already been depleted. Protein fermentation produces potentially harmful metabolites including ammonia, which can damage the gut lining, phenols, and indoles, some of which have toxic or pro-carcinogenic properties. Hydrogen sulfide, which impairs colonocyte function and may promote inflammation, and branched-chain fatty acids, which have been implicated in insulin resistance. Epidemiological studies have long found associations between high red meat consumption and increased colorectal cancer risk, while multiple mechanisms likely contribute, including heme iron, N-nitroso compounds formed during meat processing, and carcinogens generated during high-temperature cooking. Alterations in the gut microbiome and enhanced protein fermentation may also play roles. High-fat diets, especially those rich in saturated fats, are associated with decreased microbial diversity and increased abundances of bacteria linked to inflammation. Animal studies show that high-fat diets alter the gut microbiome in ways that increase intestinal permeability, promote low-grade inflammation, and contribute to metabolic dysfunction. Some of these effects appear to be mediated through changes in bile acid metabolism. High-fat diets increase bile acid secretion, and bile acids have antimicrobial properties that can alter gut microbial composition. Secondary bile acids produced by gut bacteria from primary bile acids secreted by the liver can promote inflammation and have been linked to colorectal cancer development. Conversely, diets rich in plant-based fats, particularly those containing omega-3 fatty acids from fish and certain plant sources, or monounsaturated fats from olive oil, appear to have different and generally more favorable effects on the microbiome, increasing diversity and abundances of beneficial bacteria while reducing inflammatory markers. The Mediterranean diet, characterized by high consumption of fruits, vegetables, whole grains, legumes, olive oil, and fish, with moderate wine consumption and low intake of red meat and processed foods, has been extensively studied for its health benefits. People adhering closely to a Mediterranean dietary pattern show lower risks of cardiovascular disease, diabetes, certain cancers, neurodegenerative diseases, and all-cause mortality in multiple large cohort studies. Effects on the gut microbiome likely contribute to these benefits. Mediterranean diet adherence correlates with increased microbial diversity, higher abundances of beneficial bacteria producing short-chain fatty acids, and reduced markers of inflammation. Fermented foods represent another dietary influence on the microbiome and arguably one of the oldest deliberate manipulations of microbial communities in human culture. For thousands of years, people have fermented foods to preserve them, enhance flavors, and potentially improve digestibility and nutritional value. Foods like yogurt, kefir, kimchi, sauerkraut, miso, tempeh, traditional pickles, kombucha, and countless others are created through microbial fermentation and contain living bacteria, yeasts, or both. When you consume fermented foods, you're ingesting living microbes. Do these microbes colonize your gut and become permanent residents? Generally, no. Most foodborne microbes pass through your digestive system transiently, unable to establish stable populations in the complex ecosystem already occupying your gut. But transient presence may still have effects. These microbes can interact with your resident microbiota, producing metabolites, competing for resources, affecting community dynamics. They can interact with your immune system, presenting antigens and influencing immune responses. Some studies have found that regular consumption of fermented foods increases gut microbial diversity and reduces markers of inflammation, though results vary depending on the specific foods, the baseline microbiome, and individual factors. Artificial sweeteners, ubiquitous in modern diets, represent a more recent dietary innovation with potentially concerning microbiome effects. Multiple studies in mice have shown that certain artificial sweeteners, including saccharin, sucralose, and aspartame, can alter gut microbial composition in ways that impair glucose tolerance, essentially promoting the very metabolic dysfunction they're meant to help prevent. The mechanisms appear to involve changes in microbial metabolism and altered production of metabolites that affect glucose homeostasis. Human studies have been more limited and yielded mixed results, but a controlled trial where participants consumed saccharin or sucralose for a week found that some individuals showed significant glycemic responses and microbiome alterations while others didn't, suggesting person-to-person variation in susceptibility to these effects. The long-term health implications remain unclear, but the possibility that artificial sweeteners may harm metabolic health through effects on the microbiome has prompted calls for more research and caution about their unrestricted use. Polyphenols, plant compounds that give many fruits, vegetables, and beverages their colors and some of their health benefits, also interact with the gut microbiome in interesting ways. Most polyphenols are poorly absorbed in the small intestine, and substantial amounts reach the colon, where gut bacteria metabolize them. This bacterial metabolism produces various metabolites, some of which have antioxidant, anti-inflammatory, or other beneficial properties. This represents another pathway by which plant-rich diets may benefit health through effects on and interactions with the microbiome. Even the timing of eating may matter. Your gut microbiome, like many biological systems, exhibits circadian rhythms, daily oscillations in gene expression and metabolic activity. These rhythms are synchronized with your feeding patterns and your own circadian clock. Disrupting these rhythms through irregular eating, shift work, or jet lag may have metabolic consequences, potentially mediated through effects on the microbiome. Studies in mice show that jet lag and circadian disruption alter the gut microbiome in ways that promote weight gain and glucose intolerance. Whether similar effects occur in humans, and whether interventions targeting circadian alignment can improve metabolic health through microbiome effects, are active areas of investigation. All of this raises fundamental questions about modern industrialized diets. For millions of years, human ancestors consumed diets rich in diverse plant fibers, with regular exposure to fermented foods through natural fermentation of stored foods and relatively little processed food. Modern Western diets represent a radical departure. Fiber-poor, dominated by processed foods with limited microbial diversity, rich in refined sugars and saturated fats. Are we starving our microbiomes? And if so, what are the consequences? Comparative studies of traditional societies provide suggestive evidence. Hunter-gatherer populations like the Hadza of Tanzania, who consume diets extraordinarily rich in diverse plant fibers and show remarkable seasonal variation in food intake, harbor gut microbiomes that are far more diverse than those of people in industrialized nations. They show higher abundances of fiber-fermenting bacteria and different metabolic profiles. Agricultural communities in rural areas of various countries similarly show greater microbiome diversity than urban populations in the same countries. Some researchers argue that industrialization and dietary modernization have caused a loss of ancestral microbial diversity, potentially contributing to rising rates of obesity, diabetes, autoimmune diseases, and allergic conditions in developed countries. They point out that many traditional societies show much lower rates of these diseases despite genetic backgrounds that should confer similar susceptibility. The microbiome shaped by diet and environmental exposures may be a key mediating factor. Whether and how we can restore lost microbial diversity, and whether doing so would improve health, remain open questions. But the evidence strongly suggests that diet is one of the most powerful tools we have for shaping our microbiomes, and that dietary patterns emphasizing diverse plant fibers, fermented foods, and minimally processed whole foods support microbiome configurations associated with better health outcomes. Manipulating the microbiome, the promise and limitations of intervention. If the microbiome influences health so profoundly, can we manipulate it therapeutically to prevent or treat disease? This question has spawned a massive industry, generated countless research studies, and raised hopes that microbiome-based interventions could revolutionize medicine. The reality has been more complex, with both remarkable successes and sobering limitations that highlight how much we still have to learn. Probiotics, defined as live microorganisms that, when administered in adequate amounts, confer a health benefit on the host, represent the most familiar microbiome intervention. Walk into any pharmacy or health food store and you'll find shelves lined with probiotic products containing lactobacillus, bifidobacterium, Saccharomyces, and other bacterial or fungal species. The global probiotic market was valued at over $50 billion in 2023 and continues growing rapidly. The concept is appealingly simple. Consume beneficial bacteria to improve your gut microbial composition and reap health benefits. The reality is considerably more complicated. For probiotics to work, the bacteria must survive stomach acid, resist bile salts, survive the journey through the small intestine, reach the colon alive, and either colonize, at least transiently, or produce beneficial effects during their passage. Then, they must actually influence health in measurable ways. Many probiotic strains do survive gastrointestinal transit when consumed in adequate numbers. They can be detected in stool samples while being taken, but whether they stably colonize, establishing persistent populations, is less clear. Most evidence suggests that probiotic bacteria are transient residents, detectable only while actively being consumed and disappearing soon after supplementation stops. The gut's resident microbiome, already occupying available ecological niches, seems to resist invasion by new strains in most cases. Does this transient presence suffice to produce health benefits? Sometimes, yes. The best established probiotic benefits include reducing duration of acute infectious diarrhea in children, preventing antibiotic-associated diarrhea, and reducing risk of necrotizing enterocolitis in premature infants. Certain probiotic strains have been shown to reduce symptoms of irritable bowel syndrome in some patients, though effects are modest and strain-specific. Probiotics have been investigated for countless other applications, treating inflammatory bowel disease, preventing allergies, supporting mental health, boosting immune function, aiding weight loss, improving skin conditions, preventing urinary tract infections, and many more. Results have been mixed at best for most of these applications. Some studies show benefits, others show no effect, still others show potential harms. The probiotic supplement industry is largely unregulated, with enormous variation in strain specificity, viable cell counts, and quality control between products. A fundamental problem is that probiotic isn't a single thing. Different bacterial strains have different properties. Lactobacillus rhamnosus GG, one of the most studied probiotic strains, has different effects than Lactobacillus reuteri, which differs from Bifidobacterium longum, which differs from Saccharomyces boulardii, a probiotic yeast. Even different strains within a species can have dramatically different properties. Studies using one strain cannot be generalized to other strains, yet marketing often presents probiotics as interchangeable. Prebiotics take a different approach. Rather than introducing new bacteria, prebiotics are dietary compounds, typically specific types of fiber that selectively stimulate growth or activity of beneficial bacteria already present in the gut. Fructooligosaccharides, galacto-oligosaccharides, inulin, resistant starch, and various other fermentable carbohydrates qualify as prebiotics if they meet the criteria of being selectively fermented and producing health benefits. The advantage of prebiotics is that they feed your own bacteria rather than introducing foreign strains. The disadvantage is that they only work if you already harbor the right bacteria to ferment them. If your microbiome lacks key species capable of utilizing a particular prebiotic, consuming it may have little effect or may feed bacteria that don't provide the desired benefits. Symbiotics combine probiotics and prebiotics, providing both the beneficial bacteria and the food to support their growth. The idea is synergistic. The prebiotic gives the probiotic strain a competitive advantage, helping it establish or at least persist longer in the gut. Some evidence suggests symbiotics may be more effective than either probiotics or prebiotics alone, but again, effects are strain-specific and often modest. Fecal microbiota transplantation represents a far more dramatic intervention, transferring stool from a healthy donor to a patient with dysbiosis, essentially replacing the patient's entire gut microbial community. The procedure has proven remarkably effective for one indication, recurrent Clostridioides difficile infection. Patients with C. difficile that keeps coming back despite antibiotic treatment have cure rates exceeding 90% after fecal microbiota transplantation, far better than continued antibiotic treatment. The transplant restores the microbial diversity and colonization resistance that antibiotics destroyed, preventing C. difficile from re-establishing. Encouraged by this success, researchers have explored fecal microbiota transplantation for numerous other conditions associated with dysbiosis, inflammatory bowel disease, irritable bowel syndrome, obesity, diabetes, autism, multiple sclerosis, Parkinson's disease, and more. Results have been decidedly mixed. Some studies show promising effects, others show no benefit. A few have raised safety concerns about transmitting infections or adverse metabolic effects from donor to recipient. The fundamental challenge is that we don't understand the microbiome well enough to predict which microbial communities will be therapeutic for which conditions. Unlike C. difficile infection, where the goal is simply to restore colonization resistance, other conditions likely require specific microbial functions or metabolites that we cannot reliably identify or measure. Transplanting stool from a healthy donor might not provide the specific microbial capabilities needed to treat ulcerative colitis or obesity or depression. There's also the question of donor selection and screening. What makes someone a healthy donor? Should we screen for specific bacterial species or metabolic capabilities? How do we ensure donors don't harbor potential pathogens or transmit undesirable metabolic or immunological traits? Several case reports have documented transmission of obesity phenotypes, antibiotic-resistant bacteria, or infections through fecal microbiota transplantation, highlighting the need for rigorous donor screening and safety protocols. More targeted approaches are being developed. Rather than transplanting entire microbial communities, could we administer defined consortia of specific beneficial bacterial strains selected for particular functions? Several companies are developing such products, rationally designed microbial communities for specific indications. Early results are promising, but not yet definitive. Another strategy involves postbiotics, bioactive compounds produced by beneficial bacteria that could be administered directly without needing live microbes. Short-chain fatty acids themselves could be delivered orally or rectally. Bacterial metabolites with anti-inflammatory or other beneficial properties could be purified and formulated as drugs. This approach sidesteps the challenges of getting bacteria to colonize and maintaining them in the gut, but requires identifying which specific metabolites are beneficial for which conditions. Bacteriophages, viruses that infect specific bacterial species, are being explored as precision tools to selectively eliminate harmful bacteria without affecting beneficial species. Phage therapy has a long history in some countries, particularly in Eastern Europe, but has seen limited adoption in Western medicine. With rising antibiotic resistance and growing interest in microbiome-targeted therapies, phages are experiencing renewed attention as potential tools for modulating the gut microbiome. Genetic engineering of gut bacteria offers perhaps the most futuristic possibilities. Could we engineer beneficial bacteria to produce therapeutic compounds, colonize the gut, and deliver sustained treatment from within? Proof-of-concept studies have shown that engineered bacteria can produce insulin, anti-inflammatory compounds, enzymes to degrade toxic metabolites, or other therapeutic molecules in the gut environment. In mice, engineered bacteria have shown promise for treating inflammatory bowel disease, phenylketonuria, and other conditions. But translating these ideas to safe, effective human therapies faces enormous challenges, including safety concerns, regulatory hurdles, and the difficulty of ensuring engineered bacteria behave as intended in the complex gut ecosystem. All of these approaches share common challenges. The microbiome is not a simple system reducible to good bacteria and bad bacteria. It's a complex ecological network with hundreds or thousands of species interacting in ways we barely understand. Context matters enormously. A bacterial species beneficial in one situation may be neutral or harmful in another. The same microbiome composition that promotes health in one person may contribute to disease in another with different genetics, diet, or environmental exposures. This complexity means that one-size-fits-all approaches are unlikely to work. The future of microbiome-based medicine will probably involve personalized interventions tailored to an individual's existing microbiome, genetics, health status, diet, and specific health goals. We're not there yet, but we're learning. The field is young, the tools are improving, and the potential remains enormous. The future written in microbes. Imagine a future perhaps not as distant as it might seem, where microbiome analysis is standard medical practice. Before your annual checkup, you provide a stool sample. Within days, a comprehensive report arrives detailing your gut microbial ecosystem, every bacterial species present above the detection threshold, their relative abundances, their functional capabilities, the metabolites they're producing, how your microbiome compares to healthy reference populations, and personalized recommendations for optimization. The report might reveal that you're low in certain butyrate-producing bacteria, suggesting specific dietary interventions or targeted prebiotic supplementation to support their growth. It might identify concerning abundances of bacteria associated with inflammation or disease risk, prompting preventive interventions before symptoms appear. It might guide medication selection, identifying which antibiotics would be most effective against your specific infection while causing least harm to your beneficial bacteria. It might inform cancer treatment decisions, predicting whether you're likely to respond well to immunotherapy based on your microbiome composition. This vision is no longer science fiction. The technology exists. Microbiome sequencing has become faster, cheaper, and more comprehensive. Machine learning algorithms can analyze the massive datasets produced, identifying patterns invisible to human analysis. The missing piece is not technology, but knowledge. We don't yet understand the microbiome well enough to translate microbial profiles into reliable clinical guidance for most applications. But progress accelerates constantly. The Human Microbiome Project, launched in 2007 by the National Institutes of Health, produced the first comprehensive characterization of microbial communities across body sites in healthy adults. It established baseline data sets and developed analytical methods that have enabled thousands of subsequent studies. International consortia have extended this work, exploring how the microbiome changes in disease, develops from birth through childhood, varies across populations and cultures, and responds to environmental exposures. Studies are identifying specific bacterial species and metabolites associated with specific diseases. Researchers are mapping the complex interactions between microbes and diet, between microbes and host genes. Intervention studies are testing whether manipulating the microbiome through diet, probiotics, prebiotics, fecal microbiota transplantation, or other approaches can prevent or treat disease. Animal models, particularly germ-free mice, colonized with defined microbial communities, are elucidating causal mechanisms linking specific bacteria or metabolites to specific health outcomes. The ultimate goal is not just correlation, but causation and mechanism understood well enough to intervene effectively, to identify which microbial features matter for which aspects of health, to develop targeted interventions that shift the microbiome in beneficial directions. to integrate microbiome medicine into standard clinical practice alongside genomics, proteomics, metabolomics, and other omics technologies in a truly comprehensive, personalized medicine. This will require overcoming significant challenges. The microbiome is influenced by countless factors: genetics, diet, medications, environmental exposures, age, stress, sleep, alcohol consumption, exercise, making it difficult to isolate effects of any single intervention. Individual variation is enormous, complicating efforts to establish universal standards for a healthy microbiome. Ethical questions arise around fecal transplantation, commercial microbiome testing, genetic engineering of gut bacteria, and ownership of microbiome data, but the potential benefits are immense. If microbiome-based interventions could reduce the burden of inflammatory bowel disease, metabolic syndrome, obesity, diabetes, cardiovascular disease, certain cancers, neurodegenerative diseases, allergies, autoimmune conditions, and mental health disorders, the impact on human suffering and healthcare costs would be profound and transformative. The philosophical implications. Beyond medical and scientific questions, the microbiome raises deep philosophical issues about the nature of self, identity, and what it means to be an individual organism. These questions may seem abstract, but they cut to the core of how we understand ourselves. We're taught from childhood to think of ourselves as individuals, bounded by our skin, defined by our human genome passed down from our parents. This view is deeply ingrained in Western philosophy and culture. We have individual rights, individual identities, individual bodies. We are distinct, separate, autonomous beings. But the microbiome challenges this view fundamentally. You are not a single organism, but an ecosystem, a superorganism, a holobiont composed of human and microbial cells, so integrated that separating them becomes meaningless. Your body is habitat for trillions of organisms whose genes outnumber your own by orders of magnitude. These organisms perform functions essential to your survival. They digest food you cannot digest alone. They produce vitamins you cannot synthesize. They train your immune system. They influence your brain and behavior. They are not external invaders or foreign agents, but essential components of your biology. Where does you end and they begin? The question has no clear answer. Your human cells and microbial cells are interdependent, their metabolisms intertwined, their functions integrated. When microbiome composition changes, your physiology changes. When you alter your environment or behavior, your microbiome responds. The boundary between self and other, once seemingly clear and fixed, becomes fuzzy and permeable. This challenges biological notions of individuality at a fundamental level. If your health depends on maintaining healthy microbial communities, if your metabolism is partly determined by which microbes you harbor, if your immune system requires microbial education, if your brain chemistry is influenced by bacterial metabolites, then in what sense are you a unified individual? Some philosophers have proposed that we should reconceptualize the individual organism as the holobiont, the functional unit of host plus microbiome, rather than the host alone. Others argue this goes too far, that despite intimate interactions, humans and microbes remain distinct entities with partially conflicting evolutionary interests. The debate is ongoing, but it forces us to think carefully about concepts we usually take for granted. The microbiome also raises questions about evolution and adaptation. Humans and their associated microbes have co-evolved for millions of years. Many human genetic variations involve immune system genes, affecting how we interact with microbes. The relationship isn't just ecological, but evolutionary, shaped by natural selection acting on both partners. This co-evolution means that rapid changes in our microbial environment may have consequences we don't yet appreciate. The past century has seen dramatic shifts in human microbial exposures. Antibiotics, cesarean births, formula feeding, sanitation, processed diets, urban living, reduced contact with soil and animals. These changes happened far too quickly for human genetic evolution to respond. We may be living with microbiomes fundamentally different from those our ancestors evolved with, and this mismatch may contribute to modern diseases. Some researchers argue we're experiencing a biodiversity crisis, not just in external ecosystems, but in the internal microbial ecosystems of human bodies. Just as habitat destruction and pollution drive extinctions worldwide, modern lifestyles may be driving microbial extinctions in human populations. If certain beneficial bacterial species are being lost from humanity's collective microbiome, what are the consequences? Can we preserve microbial diversity? Should we establish biobanks storing microbial strains from traditional populations before they're lost? These questions have no easy answers, but demand serious consideration. A world within. As you settle into sleep tonight, take a moment to appreciate the profound strangeness and wonder of your existence. Right now, this very moment, approximately 39 trillion microbes are living their lives on and within your body. In your gut, bacteria are fermenting dietary fiber into short-chain fatty acids that will fuel your cells tomorrow. In your mouth, bacteria are forming biofilms on your teeth, nightly construction projects that will be temporarily disrupted when you brush tomorrow morning. On your skin, bacteria are metabolizing oils and sweat, creating the chemical signature that makes you, you. In your lungs, sparse populations of bacteria interact with your immune cells, maintaining the delicate balance between vigilance and tolerance. These organisms are not passive passengers. They're active participants in the ongoing process of keeping you alive and healthy. Without them, you would not exist as you do. You could not digest food properly, develop a functional immune system, resist colonization by pathogens, maintain metabolic homeostasis, or produce essential vitamins. Your body is not a fortress keeping the world at bay, but a habitat, an ecosystem, a world unto itself teeming with microscopic life. The story of the human microbiome is still being written. Each day brings new discoveries, unexpected connections, previously unknown mechanisms. We're learning that the microbiome influences aspects of health we never suspected, from cancer treatment responses to neurodegenerative diseases, to mental health, to cardiovascular risk. We're learning that early life exposures shape lifelong health through effects on microbiome establishment. We're learning that diet is one of the most powerful tools for modulating this hidden ecosystem, but we're also learning humility. The microbiome is vastly more complex than early researchers imagined. Simple interventions often don't work as hoped. Causation is tangled with correlation in ways that make prediction difficult. The same microbial community promoting health in one context may contribute to disease in another. Every answer generates new questions. Each discovery reveals new layers of complexity. This should not discourage us, but inspire us. The microbiome has already transformed medicine, transformed our understanding of human biology, transformed how we think about what it means to be human. And we are only beginning. The hidden universe inside you is vast and largely unexplored, a frontier as exciting and mysterious as any in science. You carry this universe with you wherever you go, an invisible companion that has been with you since birth and will remain until death. It shapes who you are in ways both profound and subtle. It connects you to every other human through the shared experience of hosting trillions of microbial cells. It connects you to every ecosystem on Earth through the evolutionary relationships linking all life. It reminds us that we are not separate from nature, but deeply embedded within it, participants in ecological networks that extend from the microscopic to the global scale. Tonight, as you drift toward sleep, this microbial universe continues its ceaseless activity. Bacteria ferment and metabolize, producing waste products that become your nutrients and signaling molecules. Immune cells patrol intestinal borders, sampling bacteria, distinguishing friend from foe. Short-chain fatty acids enter your bloodstream, carrying bacterial messages to distant tissues. The conversation between your cells and your microbes, between your genes and their genes, continues unabated, a dialogue that has been ongoing since before you were born and will continue until your last breath and beyond. You are not one. You have never been one. You are multitude, ecosystem, superorganism, holobiont. You are a living, breathing example of the profound interconnectedness of life on Earth, a walking demonstration that survival and health depend not on separation but on cooperation, not on purity but on diversity, not on isolation but on community. And that perhaps is the most wonderful truth. The microbiome has revealed that we are all, in the most literal sense, in this together. If you found this exploration of the hidden universe inside you thought-provoking, please consider liking this video and subscribing to DreamLab Knowledge for more deep dives into the fascinating, complex, and often unsettling realities of our changing world. Your support helps us continue bringing you these stories that matter, these glimpses into the extraordinary transformations occurring in biology, in medicine, in our understanding of what it means to be alive, and these reminders that we are all connected to even the most invisible inhabitants of our shared existence. Sleep well, and may your dreams be as diverse and resilient as your microbiome.